At Clinexel Life Sciences Private Limited, we are your trusted partner for clinical research and pharmacovigilance. We strive to bring excellence to every project, ensuring the highest standards of quality and compliance. Recognized as the best full-service CRO in clinical trials for 2023 and 2024, our dedicated team of experts is committed to driving successful outcomes in clinical development and regulatory affairs. Clinexel also takes pride in receiving the prestigious Excellence Award for Medical Writing Services 2024, Asia by GHP UK Awards, solidifying our role as industry leaders. Clinexel Life Sciences, your trusted partner for clinical research and pharmacovigilance.
Hello everyone. It's a pleasure for me to host today the founders of SurvivX BV, Dr. Remco and Ms. Leonie. SurvivX is a Netherlands-based biotech startup, and Remco is a physician with 30 plus years of extensive experience in preclinical and clinical development of vaccines and therapeutics, especially antimicrobials. And Leonie is an analytical chemist with more than 25 years of experience in the development of pharmaceuticals and diagnostic products. Remco and Leonie, thank you very much for being here with me to share more about the survivors, the science, and the innovation behind your company. Before we delve into the science and learn about the concepts behind SurvivX, I would like our audience to learn more about the experience and impressive backgrounds of the founders. So we start with Remco.
Thank you, Deepa. Let me start by thanking you for inviting us today and giving the opportunity to tell the story of SurvivX. So we're very happy that we received your invite. With regards to your question on my background, I started quite a long time ago after my medical studies in the Netherlands we're talking about the late 90s of the last century. But I started doing research in new HIV drugs as a PhD student at the University of Amsterdam. And this was a time that HIV was still a very deadly disease. We were testing the very first new antivirals that were coming out at the time. And by the end of my PhD, HIV had turned from a deadly disease into a chronic disease that people could live with for a long, long time. So that was a nice conclusion of my PhD. Of course, I was not the only person working on that, so this was really a concerted effort of many people. But what I learned from that is that you can actually make great achievements with medical interventions in infectious diseases. So that has been sort of the red line throughout my career. I worked in vaccine development for neglected tropical diseases, parasitic diseases that actually affect a lot of people. The innovation that we're working on in SurvivX actually has a background in that research. Maybe we can touch upon that later during the interview. I'm convinced that it is very important that we stay ahead of pathogens, whether it's bacterial pathogens and viral pathogens. There's not enough work done in the world at this point in time to make that happen. So we're motivated but also proud to work on an innovation that we think can really make a difference for patients.
Thank you. Very impressive background. Working in late 90s, late 80s, I would say, and early 90s on the HIV. Probably, you could got the opportunity to work on it right from the beginning. Very, very impressive. Thank you for the introduction, Remco. And Leonie. So before Leonie starts, let me tell a little bit about Leonie. So I met Leonie, during the Innovate UK's inward AMR mission, where Clinexel as well as SurvivX, we was among the 20 non-UK companies who were selected to participate in UK inward mission of Innovate UK. so the purpose was that, we were supposed to meet the UK companies and interact with them and explore the opportunities to collaborate to support the companies that are working in AMR. And I was really impressed by Leonie's extensive knowledge and experience with the early biotech space. So over to Leonie.
Yeah, thank you so much, Deepa. So a short introduction, and so actually maybe I need to start with thanking Innovate UK, because the meetings that you referred to was extremely helpful for SurvivX. I think we have met a large amount of potential collaboration partners in the UK. So I think we would definitely be back to further explore potential collaborations over there. And we had a very nice time, Deepa. We met there, and also I understand more about your company, and you also we could be very nice collaborative partners in the future. So thank you again for this platform and having the discussion with us about our company. So a bit about my background. I'm indeed an analytical chemist by training, and right from the start, after my training, I started working at an early discovery company, the drug discovery company, where we were identifying novel entities, drug molecules from the, let's say, the bioavailable space we have. And that caught really my attention, where I was extremely interested in kind of the inventions that are made in early biotech companies that could have a very big benefit for future patients. So there I continued my career, where I was in managerial positions in many biotech companies, first of all in the pharmaceutical space, but later also in the diagnostic space. And typically I have roles of as a chief business officer or a chief operations officer, and very motivated to develop novel discoveries and bring them to the patient. So everything that is really needed to generate the evidence and really make sure that we have safe and new effective medicines for patients really motivates me to develop novel compounds. And so that's a few years ago I met Remco, and last year we had the opportunity to start our company SurvivX. So in 2023 we were founded, and since then we are developing our SUR101 platform.
Wonderful. Extensive experience indeed. Thank you, Leonie, for the introduction. So can you tell our audience about SUR101, the technology behind the foundation of SurvivX?
Yes, absolutely. So SUR101 is our lead compound. It is a recombinantly produced protein, and SUR101 also has we're developing that in multiple variants, and we will actually develop that as a technology platform of very potent innate immunity boosters. And it is able to revive dysfunctional innate immune cells. The innate immune system, I think people have heard about it, is one of the oldest parts of our immune system. And it is the first line of defense. So basically, as soon as bacteria start attacking our body, the innate immune system starts playing a role. But it also has a strong influence on the effectiveness of the adaptive immune system. And so for a long time, the innate immune system has unrightfully been underappreciated as a therapeutic target. But now the tide is turning. And we are targeting a weakness of this innate immune system, which is called immune tolerance or immune paralysis, and sometimes even called immune exhaustion. And it is actually seen and observed in many disease settings. And it's some kind of a laziness of the innate immune system after it has been stimulated for a too long time. And basically what our compound is doing, SUR101, it's the reversal of this immune tolerance. And we really think that this is a very promising emerging therapeutic potential, and in various therapeutic fields, which could be extremely relevant for larger pharmaceutical companies in the future.
Okay, great. I mean, of course, we have been, reading every day and working also on various, immunotherapeutic drugs, but targeting something right at the core or root of the problem is probably, the best option we can have here. So that's really impressive to see what you are planning. So I also understand that there is an interesting story behind the discovery of SUV101. So can you tell our audience about the same?
Yeah, I think it's very interesting to mention that our lead compound was discovered in an observational study in India. So researchers in the field of parasitic disease really found that people who are infected with a roundworm parasite are less susceptible to sepsis. And this work was done several years ago, so more than 10 years ago, by Professor Ravindran in India. And well, there was a huge interest to understand what is causing this protective effect. So their group continued to further investigate and actually found that the parasites are secreting a protein that is basically protecting the people for sepsis. And what they have found in the years thereafter is that it is a protein that is belonging to the heat shock protein family, and it is activating toll-like receptors 2 and 4, and possibly also working via the CD14 receptor. And so this is what we know now about the compound. The organization CCAMP has outlicensed the technology and the IP to SurvivX. So we are very happy with that. And we now want to further develop this as a molecule, as a pharmaceutical entity.
Wow, really interesting. An observant physician who could, who observed that patients who are suffering from roundworm infection, they are susceptible to sepsis. Actually, sepsis, around 15% of admitted patients, there is a risk of, developing sepsis. That's really interesting. And then probably more scientific discovery was done around it to figure out, what may be the cause. And I'm really impressed to see how this, worked around. So wonderful. And the discovery was done in India. That makes me all the more happy. With the, together with Dr. Ravindran and CCAMP, really good job by CCAMP and Dr. Ravindran. So, so being an immune booster, this SUV101 therapy is expected to have therapeutic potentials in multiple indications. Is this correct?
Yeah, that's correct, Deepa. And let me first start answering a remark that you just made, that so many patients are being admitted in hospital and develop at some point in time sepsis. And I think this is something that is still underappreciated. People know what to do when they have pain on the chest, for example. They know that they need to go to the doctor because it's an emergency. But with sepsis, symptoms are sometimes overlooked. It's not immediately recognized. So there's a lot of benefit that can be gained in the prognosis of patients with sepsis, both on the front of the identification of patients with sepsis, so making sure that they end up in the hospital early enough, as well as making sure that we can address the different phenomena that we see with sepsis. And like Leonie already mentioned in the beginning, we work on a particular weakness of the immune system that especially becomes apparent when patients are admitted with sepsis and already have developed sepsis for a couple of days. Then you see that the immune system sometimes gets, Leonie called it laziness, you can also say it's overwhelmed, it's becoming tired, and that's the phase where you actually need to do something, and we believe that our molecule is actually better than what is available on the market today, which is really limited what doctors have in hand. Like Leonie mentioned, our molecule works on what we call the innate immune system. So it's a different part of the immune system compared to the adaptive immune system that a lot of people are familiar with antibodies and cellular responses. And the importance of the innate immune system has sometimes, or at least in the past, been overlooked. So people really felt this was sort of an older part of the immune system, but it was not discriminatory enough against different pathogens. But we have learned over the last 10 years or so that the innate immune system is really tremendously important on its own, but also to support the adaptive immune system. And how it works in research is sometimes that you have to study your data over and over again to see what is actually happening. So when we, learned first about this project, when we met Prof. Dan, already saw some of the impressive data on what the molecule does on the innate immune system. When we established SurvivX in 2023, we also started collaborating with researchers here in the Netherlands. So we work with the two top universities that do research on the innate immune system and the development of sepsis. And in those experiments, the researchers actually told us, hey, we see very remarkable results, and we believe it's better than the compounds that are out there on the market at this point in time. And that made us realize that if that principle is true for sepsis, it may also be true for other diseases as well. So to make a long story short, the innate immune system can play a critical role in the treatment of cancer, for example, where for the last 10 years we have seen actually impressive new results from new strategies ranging from checkpoint inhibitors to CAR T cells to other form of cell-directed therapies, where in some cancers that works actually very well, and in others it doesn't. And the innate immune system is probably playing a large role in that, and we think that the innovation that we have in hand may actually help the innate immune system to make sure that those treatments become more effective. Likewise, there are other diseases where that phenomenon that the immune system gets tired becomes more prominent. You, for example, see that in patients that are admitted to the ICU for a long period of time. You see that in major surgery. So these are all indications where we believe our molecule could play a role. To close my answer to your question, are we certain about this? No, we're not. We're in very early stages. So, we get to talk about our plans hopefully a bit later. But for us, it's now crucial to make that step to see, we've proven in preclinical studies that our molecule is potentially highly effective in the setting of sepsis. Can we also replicate that with early research looking at these other indications?
Oh, that's really interesting. In fact, the indications that you are targeting with SUR-101, there is a huge therapeutic unmet need in these indications. In fact, 20% of deaths worldwide are caused because of sepsis. So, and cancer, we know how the incidence is rapidly increasing and the role that immunotherapy is playing. But one of the challenges that we see when we conduct the clinical trials in sepsis, or for that matter in oncology, the heterogeneity of the patients, that becomes challenging. So if we can, identify in the beginning who, while you are doing your in vivo and vitro studies, who are the patients who will be potentially benefited, with this, so that we can, address this part and can have probably better clinical data when you are in clinical development.
Exactly, and what is very encouraging is that this summer there was a presentation of the very first study in sepsis patients where a personalized strategy was tried. And the personalized strategy meant that people who were admitted for sepsis underwent a blood test early on to differentiate two stages of sepsis. The first stage is the stage of hyperinflammation, so that is the stage where your immune system starts to respond to the pathogens, and that is a stage that people know quite well, because these are the typical symptoms that people associate with sepsis. But there's also this phase of immune exhaustion that we now discussed 2 times in answering your questions. And you can actually use blood tests to actually differentiate for that. So the hyperinflammation state is something that you can detect with a ferritin test, and the hypoinflammation state, so the stage of exhaustion, is something that you can measure with a test called HLA-DR. So you test for a particular receptor on cells of patients, and that's very indicative whether patients are in a hyperinflammation or in a hypoinflammatory state. And the nice thing about the study that was presented over the summer, which is called the immuno-oncology study, was that it showed for the first time that if you have a specific treatment for the hyperinflammatory stage versus the lower inflammatory stage, that you can actually get a better prognosis for patients. And this was the first time in a clinical study that they showed a statistically significant difference. Now, the reason why we believe that our molecule is could be more successful in patients is that this study was done with a compound called interferon gamma. This was the molecule that was used to treat patients with a hypoinflammatory state. And if you look at the in vitro work on our molecule in comparison with interferon gamma, you can actually see that our molecule performs better in those tests. So we have good hope that if we can repeat that in animal studies and later in clinical studies, that we actually can get a better prognosis of patients.
That's perfect. That's what would make it its therapeutic potential really higher as compared to the existing drugs or other one. If you precisely know which patients are going to be benefited, that will add a lot of value. And that's what I was hoping to hear. Sorry. Yes.
And maybe, Deepa, maybe I can also add here, if you look at the additional indications, is that what we have seen is that the immune suppressed state in sepsis is basically occurring independent of the type of infection. So it could be bacterial infection or it could be a viral infection. And what we have learned over the last few years is that also people who died from COVID-19 on the intensive care units actually died from sepsis. And this is something that a lot of people do not realize. And within the patient population, amongst those patients, 30% of the patients does have an immune suppressed state. So we believe that also our molecule could be very important, let's say, therapeutic agent for a general preparedness in future pandemics. So maybe that is also a very important indication to look at in the future.
It's interesting. So what kind of commercial market you are targeting, the size of the market?
So what we're currently want to investigate, so of course the sepsis market is very large. It's let's say for pharmaceutical companies it will be let's say around $6 billion US dollar value. But we are also currently exploring other indications, so like the immuno-oncology space, which is extremely large. And if you look at the use of our compound, we could use it to potentiate basically the current and maybe even failing immuno-oncology therapies. But we can also use it as a standalone immuno-oncology therapy where we can really boost the immune system to really start attacking the tumor itself. So those are really, really large indications.
It's interesting. Wonderful. And who have been your partners so far in this journey?
Yeah, so of course it all started with CCamp, and we're very, very happy with the very nice and collaboration together with the CCamp team, but also with Professor Ravindran, who is always on the background and supporting us. And we have very close relationships and multiple and frequent meetings with them. And of course, the research is continuously ongoing. But of course, we also work together with the Radboud UMC in Nijmegen in the Netherlands. And they were very happy to learn about the technology. So the Department of Experimental Intensive Care Medicine really said, okay, we really would love to test this compound, just and see in our own hands how this compound works. And they, every time we met, I was very pleasantly surprised by their surprise that the compound really is showing what it's doing, what it should be doing. So they kind of validated independently the effectiveness of the compound, and that's why we are now so, let's say, positive about the future of the compound. So apart from as Radboud UMC and CCAMP, of course, we also collaborate with Innate, who have very kindly supported us with a small grant in 2023 when we started. And we, of course, want to continue our collaborations with the Innate network. And there we also received, when we started our company, a convertible loan from OostNL, which is a regional investment fund, so who we also closely collaborate with. And so we would like to continue our development, and once we have more investments, then we will, of course, start working together with multiple partners that we have aligned for the future development of our compounds.
Mm-hmm. I'm sure pharmaceutical companies as well as investors would be very much interested, to collaborate with you. And in fact, Professor would also be very happy, Professor Ravindran, to see, that his invention is in good hands and moving forward properly. So can you highlight a little bit more for the interest of the audience, what sort of data, in vitro and in vivo data, that you have already generated for the compound?
Yeah, thanks. That's an excellent question. We have done both what we call in vitro studies as well as in vivo studies. So in vitro studies are studies in the lab where you actually look in test tubes what the effect of your molecule is. And in vivo studies are typically studies that you do in animals to see whether a molecule has a certain effect. And we're very happy that the in vitro results that we have generated showed that our molecule is really capable of completely reverse this state of immune suppression. So it's really a very powerful stimulant of innate immune cells, and I think I already mentioned that we do better than other compounds that are currently on the market. But I think it's very important that we consider that to be a very key result, because it is for us the key to potential use in other therapeutic areas as well. But the proof of the pudding, of course, is always in the eating. So that is what you try to establish in the animal studies. So we have done two studies where we looked at both bacterial sepsis as well as in viral sepsis. Those are two models that are being investigated in mice. And actually in both models, we actually showed a survival benefit. So the animals that were treated with our molecule had a statistically significant better survival than the animals that didn't. So that is for us a very important proof of principle. It's not enough yet to go into clinical studies, so there's still a lot of work that needs to be done. But we are confident based on these data that our molecule has potential. And especially in the context also with what we learned this year of the fact that personalized medicine in the setting of sepsis is actually something that works. We're very confident that this is the way to go forward.
Interesting.
Maybe to add here is that, of course, you can say, okay, animal models, how representative is this? Of course, this is our first indication. But what the people at Radboud UMC also did is taking, let's say, white blood cells from healthy volunteers, and then put them into immune paralyzed state, and they were fully able to reverse that paralysis in the human cells. So for us, so it's not happening in the body, but actually in the test tube, but it really was with human cells that were immune suppressed. So this is also something that is for us the bridge where we could say, okay, we see the potential use in humans.
Yeah, for sure. Of course, everything starts, all the new molecules first, they start from in vitro and then eventually in vivo, and then you will, after the IND enabling studies, you will be at a stage to conduct the clinical trials. So that will be eventually lead to the approvals. But it has been a good start. In just a year, you have generated so much of data. You already have good partners from CCAMP to INCATE to UMC. Wonderful, impressive in vivo and in vitro data, and really good partnerships and collaborations. Really impressive. No wonder Innovate UK, selected you for their Inward UK mission. There were only 20 companies, non-UK companies selected for that. So it's really impressive what you have done in a year. So what are your next plans?
Yeah, thank you for the compliment, Deepa. We are proud of what we have achieved over the last year. At the same time, it's just the beginning of a journey. This is a call to action for the audience that is watching. If you're interested, if you're a pharma company representative, if you're an investor, contact us. We have a very simple email address, info@survivx.com. We're happy to talk. And Leonie can perhaps tell a bit more on our future plans.
Yeah, thanks, Remco. So our future plans are basically two ways. So what we prefer to do is to further develop this compound and develop it until a phase 1, phase 2 clinical trial setting. So where we would like to demonstrate the efficacy as well as the safety of the compound. And then we would like to out-license to a larger pharma partner. The compound can be developed, of course, in multiple indications, like we said. So what we would like to do in the, let's say, the near future is work on, identifying, doing multi-omic evaluations to really understand the mechanism of action better. And we would like to understand more about the biomarkers that are driving our therapy, but also identifying the patients that are most susceptible and most likely to respond to our treatment. But we would also like to evaluate in multiple animal models, in multiple indications, where our molecule is currently the most effective, so that we can identify the priority indication where we would like to start doing our first clinical studies in. So that could be immuno-oncology, it could be, other indications, it could be sepsis. So currently we are in a stage of raising a seed investment round of 3 million euros to do all that work. And then we would like to continue to prepare ourselves for our clinical trials in that priority indication. So of course we would like to out-license after our clinical proof of concept stages, but we are also open for early out-licensing or early co-developments with pharmaceutical companies or biotech companies, because with joint forces, of course, we know we can make maybe even faster results.
Great. Very interesting. That's what I like about the small biotech, the dynamic approach as well as the flexibility, like you have mentioned, that you are open for investments as well as collaborations at an early stage, or you are open to take it up forward through, the preclinical and then early stage clinical development also. And definitely, I am pretty sure there would be a lot of interest from pharma companies on, talking to you. So definitely, I think there will be some, opportunities coming up on your way. Any concluding remarks that both of you want to share here?
Well, for me, I would like to first thank you for the opportunity to have this interview. It's really important that we tell our story and that the world gets familiar with what we have to offer. I think it's already mentioned the email address, but I will mention it one more time if people are interested to talk to us. info@survivix.com.
Leonie, anything from your side?
Also, again, thank you so much for the opportunity for presenting our company today, and really love to work with you in the future if possible.
Thank you so much to both of you, Leonie and Remco, for being here. It's wonderful interacting with you. Take care and have a nice day ahead. Wish all the best to you as well as SurvivX for the future success.
Thank you so much.
Thank you.
Thank you. Bye-bye.
Bye-bye.
At Clinexel Life Sciences, we deliver innovative solutions. Join us in shaping the future of healthcare with precision, dedication, and trust. Thank you for choosing Clinexel, where excellence in clinical research is our promise to you. Clinexel, your trusted partner for clinical research and pharmacovigilance.